For a long time, it has been widely believed that learning and memory are only related to the brain. However, a latest research has found that other cells in the body can also learn and form memories, which has opened up new avenues for understanding the formation of memory and brought new possibilities for treating diseases related to memory. The research results were published in the journal Nature Communications on October 7th. In the field of memory and learning, spaced learning and massed learning are relative concepts. Spaced learning refers to dispersing the learning content and conducting it over a period of time with a certain time interval between each learning. This method helps us repeatedly receive information at different time points and converts short-term memory into long-term memory. On the other hand, massed learning means continuously and intensively learning in a short period of time. Although this cramming learning method may seem more effective in the short term, in the long run, this single and intensive learning process cannot retain information more effectively because the key to memory and learning is repetition, whether in a waking state or in a dream. A large number of past studies have shown that spaced learning can promote the connection and remodeling of brain neurons, thereby strengthening the formation and consolidation of memory and improving the persistence and accuracy of memory. In the experiment, they studied two types of non-brain human cells, one from neural tissue and the other from kidney tissue. The researchers exposed them to different patterns of chemical signals to simulate the learning process over time, just as brain cells are exposed to neurotransmitters when we learn new information. In response, kidney cells also produce the same pattern as brain cells and turn on a memory gene. To more intuitively monitor the memory and learning process of these cells, the researchers modified them to produce a glowing protein that can show when the memory gene is turned on and off. The study found that these non-brain cells can also recognize the repeated patterns of chemical pulses used to simulate the bursts of neurotransmitters in the brain, and the memory gene, activated by spaced pulses, is stronger and more persistent than the same number of single pulses sent at one time. This phenomenon is very similar to the way neurons in the brain learn and remember, indicating that non-brain cells also have the ability to learn and remember. This research shows that the learning ability is not unique to brain cells. In fact, it may be a basic property of all cells. A case report published in the journal Vaccines on August 1st outlined how a female scientist successfully treated her own stage 3 cancer by injecting a virus she cultivated in the laboratory into the tumor, and she has now been in remission for 4 years. Oncolytic virus therapy is an emerging field in cancer treatment that uses viruses to attack cancer cells and stimulates the immune system to fight cancer cells. However, currently there's no OVT drug approved anywhere in the world for the treatment of breast cancer at any stage. She is not an expert in this area herself, but her professional knowledge in culturing and purifying viruses in the laboratory gave her the courage to attempt this treatment method. She chose to use 2 different viruses in succession. One is the measles virus, and the other is the vesicular stomatitis virus. Both viruses have a good safety record. The measles strain has been widely used in children's vaccines, and the VSV strain would only cause mild flu symptoms in the worst-case scenario. A colleague injected the virus she prepared directly into her tumor. After that, the tumor shrank significantly, became softer, and there were no serious side effects. It also detached from the invaded pectoral muscles and skin, making it easy to remove surgically. The analysis of the resected tumor showed that the tumor had been completely infiltrated by immune cells, indicating that the OVT therapy had played its expected role and stimulated her immune system to attack the virus and tumor cells. Her experience did not break new ground because this method had been tried before. What made her remarkable was that she tried 2 viruses in succession and cultured the viruses herself in the laboratory. Perhaps due to considerations of self-experimentation and ethics, the paper was rejected by more than a dozen journals. Maybe the problem is not self-experimentation, but that the research results she published may encourage others to try similar methods. Therefore, the author also emphasizes that this self-therapy should not be the first choice. Observational data from the James Webb Space Telescope shows that a black hole appears to be devouring matter at a rate 40 times faster than the Eddington limit. This discovery provides valuable clues for understanding the rapid growth mechanism of supermassive black holes in the early universe. The related research was published in the latest issue of Nature Astronomy. This black hole, named LID 568, only 1.5 billion years after the Big Bang. By comprehensively observing the target galaxy and its surrounding environment, the team unexpectedly discovered a powerful gas outflow phenomenon around the central black hole. Based on its speed and scale, it is speculated that its mass growth may be achieved through a rapid accretion process. This ultra-fast feeding mechanism beyond the limit may be the reason why such heavyweight black holes appeared so early in the universe. These findings provide a new perspective on the formation of supermassive black holes. They may either come from the death of the first batch of stars in the universe or be directly formed by the collapse of gas. Now the discovery of super-Eddington accretion black holes means that regardless of the formation method, they may all enter a rapid accretion event and experience significant growth. Space junk is composed of the debris of old rockets, satellites, and other objects launched into orbit since 1957. Currently, there are approximately more than 40,000 objects with a diameter greater than 10 centimeters, 1.1 million objects with a diameter between 1 and 10 centimeters, and 130 million objects with a diameter between 1 millimeter and 1 centimeter in low Earth orbit. With commercial space companies continuing to deploy giant satellite constellations for research, telecommunications, and internet services, this situation will further deteriorate. To solve this problem, researchers at Kyoto University have developed the world's first wooden satellite. Except for the electronic components, this small satellite is entirely made of wood. This satellite will burn up completely when it re-enters the Earth's atmosphere at the end of its service life. The side length of this small satellite is 10 centimeters, and its weight is only 900 grams, making it one of the lightest satellites ever launched into space. After the satellite arrives at the International Space Station and is deployed, it will operate in space for 6 months. The ground will continuously monitor whether there are signs of deformation of the satellite. The ultimate goal is to determine whether the wooden satellite can withstand the extreme conditions in space. There is also a double-unit CubeSat planned to be launched in 2026. These are 2 elephants at the Berlin Zoo. The one on the left is called Mary, and it is using a hose to take a bath skillfully. The one on the right, perhaps thinking that Mary has been taking a bath for too long, deliberately blocks the water flow with its trunk. Why can elephants use hoses proficiently? One possible explanation is that the hose is similar to an elephant's trunk. But it is not clear how Mary acquired such excellent hose skills. None of the other 4 elephants in the zoo have similar abilities. In addition, to investigate whether Mary's companion deliberately blocked the water flow, the researcher repeatedly showed it 2 hoses, but only one was for Mary. The results are inconclusive. It often chose the hose closest to Mary, but the team said that they suspect that this interference may be deliberate and that its operation has become more effective over time. The research results were published in Current Biology on November 8th. Whether Mary uses the hose depends on the diameter of the hose. This may be because the smaller the diameter of the hose, the more difficult it is to operate. Countless animals, from chimpanzees to crows to fish, can use tools. Elephants can also use their trunks to grab leaves and use them as things like fly swatters.